So right now we're looking at frog's blood. That dark dot in the middle of all those circles is the nucleus. You can't really make out any of the other organelles, but you can see the plasma membrane around it. And remember the difference between animal and plant cells. This is an animal cell, so it doesn't have something that all plant cells have. Yeah. I'll just pan over it for a bit so you can have a look. They're all pretty similar. So that is frog's blood cells. Now I'm going to show you a different animal cell. I'll show you human blood next. And we're back. How quick was that? So this slide is a pre-made slide of human blood. Now, there's definitely a difference. This is on the same magnification as the frog's blood. And you can see that human blood cells are a lot smaller. And I'm just trying to focus it a bit more. No, that's as good as it gets. And we can't really make out any of the organelles. You can see how there's kind of a dot in the middle of some of them, which I don't think is the nucleus. I think that's just sort of showing its shape. You know how red blood cells have that weird like round shape with like a dent in the middle. But you know, the similarities between the human blood and the frog's blood, there are some similarities that I think you can notice. Okay, I'm going to show you some human skin next. All right. Ooh, would you look at that? Now you can clearly see a lot going on here. It's kind of hard to decipher, but one thing we can make out, I'm just going to move it a little bit, is those dark dots, which I would assume are nucleuses. Nucleuses are the main organelle that we can see through a microscope. So a lot of these cells, like skin cells, they're quite long and skinny. So, sorry. Let me get it back. Ooh. There we go. Sorry, gentle viewers. There we go. So you can clearly see those nucleuses, and you can see the kind of flatness of the cell. So that's like the top of the skin surface. And you can see those dark parts that are flattened out. They're actually nucleuses of long, skinny skin cells. Now this is below the skin surface, a lot of muscle cells, and you can see all this tissue bunched together, and all these dark bits again are the nucleuses of cells. Now because they're animal cells, they don't have a cell wall, so they kind of tend to be all less organized than plant cells. Ooh. Where is it? Oh, here we go. This. Okay, now this area, can you recognize those cells right there? Do they look like something that we have just viewed? If you said human blood cells, you are correct. So this cavity right here is a blood vessel, like a vein or an artery or something like that. And, oh my gosh, sorry. And all those little cells in it is the blood flowing through. So that's pretty cool. Keeps going out of focus, sorry guys. All right, now these are some plant cells of Lilium, which is a type of plant. Now you can see this has been dyed with a couple of things. That's why the nucleuses look so dark. Those pink circles are the nucleus, which contains all of the genetic material. And you can see the plant cell walls. All right, I'm just going to take you for a little bit of a tour through this plant cross section, so you can clearly see some cells all bunched together there. So this is from a stem, so it's cross section from a stem. So this is on the outside of the stem, and then it's been sliced, and we're looking down the stem, and this is the middle of the stem where the cells are reproducing, and that is actually pretty outrageously awesome. That is mitosis happening right there. I don't know if you've learned about mitosis, but that cell, this cell right here is about to split into 2 cells, and you can see the genetic material has split to go to either side, and it's about to pinch in the middle and will turn into 2 cells. So this is the middle, the inside of that stem where all of this cell division is occurring. And then when we go out, you can see more plant cells, all very organized, stacked next to each other. This is the outside of the stem. You can clearly see the nucleus in there. It's kind of difficult to make out other organelles though, but I'll show you another plant cell and we might be able to make out some organelles. Okay, now we are looking at a leaf epidermis. So this is the outside of a leaf. You can see these cells are quite long and skinny, but all very neatly packed together. See that? It's one hugely long cell, and then you've got these little round cells kind of in between. So this thing right in the middle of my screen, that oval, that oval is called a stomata, which is, this is just where the gases exit and enter the leaf. Now the stomata is made of 2 cells that are kind of hugging each other, which is why you can see 2 nucleuses and the 2 cells forming that little circle. But all the other cells are the long kind of flat ones that are packed together. All these long cells look very much like onion cells as well. And a lot of plant cells have this kind of look to them, this long, skinny, organized, tightly packed. So there's some more nucleuses that you can make out. Again, we really can't see other organelles, but that's normal with a light microscope. Okay, the last plant I'm going to show you, I am going to make the slide myself. So you can watch me do that. Hello, year 8. So we've looked at some cells that were all pre-made slides like this. See, it says human blood. So that's what I was looking at for human blood. It wasn't my blood. And like the frog's blood, they're all pre-made slides. But for the last plant, I'm going to make the slide myself. So I have taken some Alodia from the fish tank, which is this water plant. Alodia is so good to look at through a microscope because you can just get one leaf and you can see right through it. And we're going to be able to see more than just the nucleus, I promise you. So I've got my slide. I'll just turn this down so you can see me making it. Got my slide. Going to grab my Alodia, take one leaf, place it on the slide. There we go. Just going to put a drop of some water on there. With my hand. There we go. This is called a wet mount. It's called a wet mount because I am mounting a specimen onto a slide and I am adding a little bit of wetness. Now, it's not finished yet. I need to put a cover slip on top. So these are the cover slips. We've got microscope slides, which are quite a thick piece of glass, and then the cover slips are ridiculously tiny and they just break all of the time. So let's see this. But these are really, really thin pieces of glass. Just one tiny square. Can you see that? And I'm going to gently lay that on my slide. Now the way to do this is you lean it on one side and then gently push it down. Now, can you see that? There's my beautiful leaf. Now we're going to look at it under the microscope. Okay, so this is the microscope I'm using. I'll take out my leaf epidermis, which is what we just looked at with the stomata. Putting my slide onto the stage, which is this part of the microscope. It's the stage where the performance happens. And now I'm going to show you what it looks like. Okay, there we go. Oh my gosh, can you see that? Okay, so what we have here is a bunch of cells all tightly packed. One leaf is about 3 cells thick, so that's why you've kind of got this. See, if I change the focus, you can focus on different layers. And see, then that's, yeah. So we're going to try and just focus on one layer. Can you see some movement happening in the middle here? That is actually the chloroplasts moving. The chloroplasts move around. Actually, it looks like a mite. Maybe that's why my Alodia isn't surviving. It's infested. Whoa, can you guys see that? There's like little bugs inside the cells. Oh my god. So all these green dots that you can see are chloroplasts, which are only in plant cells that photosynthesize. Now the chloroplasts do move. You can see them. See down the bottom there? They're moving. Oh. All right. Now look down the bottom of the screen in the middle. Can you see those chloroplasts moving? That is called cytoplasmic streaming. So they just kind of flow around the vacuole in the middle of a plant cell. Now, if you've seen plant cell diagrams, you will know that plants have vacuoles, which is a big fluidy sac. Now, in this cell, you can't actually see the vacuole, but it's sort of implied it's there by the way the chloroplasts are dispersed. They all move kind of around this kind of empty space in the middle. It's not actually empty. There's a vacuole there that you can't really make out, but that's why they're moving the way they're moving. But can you see those mites? There are mites in there. And I have been wondering why my Alodia is not growing. And I think we have figured it out right now. There are actually tiny mites inside the cells. It's mind-blowing. There you go. There's some more cytoplasmic streaming with the chloroplasts. Oh, it's so beautiful. Looks like those mites are inside the chloroplasts, eating them. Oh my gosh. Amazing. I could stare at this all day. There's another area. Oh, there's a lot of movement here. Those tiny little mites and the chloroplasts are moving. Can you see in the middle? Got some cytoplasmic streaming. Beautiful. Okay. Thank you for watching. I hope you learned stuff.